Hi, my name is Joel Nitta. Thanks for coming to my talk, an open and continuously updated Fern Tree of Life, or FTOL. And you can find the slides from my talk online at this URL, joelnitta.github.io/botany2022 FTOL. So phylogenies are essential to the study of evolutionary biology. I think this is nicely summed up in this quote from the APG website. Only with a phylogeny can we begin to understand diversification, regularities, and patterns of evolution, or simply suggest individual evolutionary changes within a clade. And certainly, the APG taxonomic system would not be possible were it not for many large angiosperm phylogenies. So clearly, having a well-sampled phylogeny is really important. Fortunately, these days, there are many software tools that enable us to build large trees from GenBank data. And this is an example of some recently developed pipeline tools. However, any such tool does encounter some problems. So the first problem is that there is always going to be a trade-off between scalability and accuracy. So any automated pipeline must make some shortcuts and assumptions because of the large amount of data that it's working with. On the other hand, manual inspection of all sequences would be expected to lead to higher quality results, but this approach simply does not scale. Another problem is that any tree built with these sort of methods will quickly go out of date, and that is due to the rapid accumulation of data on GenBank. So when we attempt to assemble a really large tree, we need to keep that in mind. So the approach that we've taken in this project is to combine automation with customization. And this is in pursuit of the goal of constructing a pipeline to generate a maximally sampled, high taxonomic quality phylogeny of ferns. So why ferns? Well, ferns are a large, diverse, ecologically important group of plants. They happen to be in my study system, so I'm really excited to try and build a large fern tree. From a more practical standpoint, if we compare them to other similar organisms, such as seed plants or angiosperms, it turns out ferns are just an easier group to work with because of their overall lower species number. So ferns have about 12,000 species, of which maybe 40 to 50% have been sequenced, in comparison to seed plants with over 300,000 species and probably less than 20% sequenced. So ferns are just a more tractable study system to try to build a maximally sampled tree. That's not to say that other people aren't working on a maximally sampled seed plant tree, which is really important, but here we're focusing on ferns. Another important thing to understand about ferns before proceeding further is that for the past 30 years or so, Sanger sequenced plastid genes have really been the workhorse of fern molecular systematics. And you can see that in this chart, which shows the number of fern species in GenBank by the genomic compartment of the sequences. So we can see that the total number of species is almost completely represented by plastid sequences. So plastid sequences are going to be our data of choice when it comes to building a maximally sampled fern tree. Okay, with that, I'll move into the methods. So all of our data come from GenBank, and we have two broad categories of GenBank data. The Sanger data include 7 commonly used loci for fern systematics. The breakdown of that is 5 protein coding genes, and then 2 spacers. And this altogether currently accounts for about 5,100 species. The other category of data are full plastomes, and that includes 77 single copy genes and then the 2 same spacers. And of course, this is far fewer species. To use the GenBank data, we don't actually query GenBank live online, but instead I download all of the data at one go from their FTP server and then use that to create a local database with the rest-as R package. And by the way, I'm now maintaining that R package, so if anybody wants to do something similar, please feel free to ask me questions. And the reason for that is twofold. First, it increases reproducibility because now we have a fixed version of GenBank that won't change day to day. And secondly, it is also more efficient to query large amounts of data locally instead of online. Next, we use a software package called SuperCrunch to extract sequences from the accessions without relying on annotations. And that's really important because there's no standard format for how to annotate genes in GenBank. So this way we don't have to worry about various different annotation formats. After we've extracted the sequences, the next step is to resolve taxonomic names. And this is really important because the NCBI taxonomy was not designed with ferns in mind, and it includes many fern synonyms. So to do that, we start with the World Ferns database maintained by Michael Hassler. And however, the World Ferns database, although it's great because it has a lot of synonyms that we can use for resolving names, it's actually not quite set up for this specific purpose. So, and it needed some updating. So we made those changes and then released that as a taxonomic database called Pteridicat, which is compatible with our tree. We then resolved GenBank species names to the names in Pteridicat using the taxostand R package. And quick plug here, I am the developer of that R package, and I have a talk about it at Botany. So please check that out if you have the chance and you're interested in biodiversity informatics and herbarium digitization. And I also really want to stress that anybody else who wants to use our tree and our data, you can also match species names to it using these two R packages, taxostand and Pteridicat. Okay, the next step was to do some automated cleaning. And that's because, of course, there could be misidentifications in GenBank data. So to do that, we used an all-by-all BLAST approach. So we first constructed a BLAST database, including all of the sequences, and then we queried each sequence in turn against it. And any sequence that matched to the wrong family, we then flagged and excluded as a mis-ID. And we found about 70 sequences in total that needed to be excluded this way. Okay, after all of these data extraction and cleaning steps, we finally get to the phylogenetic analysis. We do that in two steps. First of all, we infer what I call the backbone using just the plastome sequences. So we align sequences with MAP and then do the phylogenetic analysis in IQ tree. The next step is to move on to the full tree. And the alignment and analysis and phylogenetic analysis is similar. The key difference being that now we use the plastome tree from the last step as a constraint. And the reason for this is that that plastome tree has really good resolution along the backbone, so we can resolve deep nodes. But then with the Sager sequencing, that also allows us to maximize sampling at the tips. Another feature here is that before we actually did the final ML analysis, we did one run of IQ tree in fast mode. And this gives me a chance to take a look at the initial analysis and try to find any more rogues that may have fallen through the cracks from our previous steps. So we'll remove those and then run the final analysis afterwards. And once the final ML analysis is done, then we date the tree using TreePL. This is an overview of the complete workflow. And the workflow is automated with the targets R package, so we can easily repeat this on a regular basis. It takes about 10 to 14 days total to run. Most of that is occupied by the ML analysis. And you can see there's some steps here with the hand. Those are places that include manual inspection to make sure we have a high quality result. Okay, moving on to the results. So this is the Fern Tree of Life, or FTOL, version 1.1.0. It's greatly increased sampling. So we have, compared to previous studies, we have 5,500 out of about 12,300 species. So about almost half of all ferns. And that's a 40% increase relative to the previous largest phylogeny. We also, this is to show that we have high bootstrap support across the tree. So the bootstrap values are higher in yellow. So you can see we have high support across the tree, and especially at some of the deeper nodes. Now, one interesting result is that when we did the divergence timing analysis, we were able to use an expanded set of fossils, 51 fossils, which is about 2 times more than any previous study. And because of that, actually, our estimated ages push back the stem ages for most families around 10 to 30 million years. And this would suggest that perhaps ferns did not actually diversify in the shadow of angiosperms, as has long been contended. And this is a plot showing the stem ages of various families in comparison with previous studies. And you can see that our ages, the red dots, tend to be older for almost all of the families. Now, I should put one caveat on this, that this is still a somewhat preliminary analysis done with TreePL. So in the future, it does need to have some more rigorous analysis to verify that this is really what's going on. Okay, so one really important thing that I can't stress enough is that we want to make our tree available for other researchers to use for their own research. And so for that, we've released it on a web portal, fernfi.github.io. You can go here, click on the Downloads tab, and download the zip data. Another really nice thing that we've implemented is a data explorer that allows you to look into more detail. So I'm just going to demo that really quickly. It's a Shiny app built in R. And if you are using the tree and you notice a species that maybe seems to be out of place, then I would encourage you to come look it up in our FTOL Explorer. You can select species on the left side, and then it'll show you information such as the accession that was used and the original taxonomy and the name that it was matched to, etc. And we're going to try to add some more functionality in the future too, to make this even more useful. Furthermore, we've also made FTOL available through an R package called FTOL-R, so you can read it directly into R. You can also load the alignment data, and you can apply various customizations, such as dropping the outgroup, etc. Again, I really want to stress that we want to make this resource available to the community and get community feedback. So one example of that is in the family Thelipteridaceae. Between two previous versions, 1.1.0 and 1.1.0, 1.00 and 1.1.0, we noticed that there are a lot of non-monophyletic genera in Thelipteridaceae. So we reached out to Susan Fawcett, who's an expert on this family, and with her input, we were able to reduce that number from 16 to 7. So we think of this as a living project, and we would love to have input from other specialists in different fern groups, and hopefully make it a better tool for everybody going forward. Okay, so in summary, I think that FTOL hits a sweet spot between automation and customization. Our workflow uses automated versioning of GenBank data, versioned mining of GenBank data, so it's fully reproducible. But we also incorporate a custom taxonomy and input from taxonomic experts. So it's a really high quality result that you can use immediately for your own research. And I hope that perhaps some of the methods that we've used can be used as a model for other plant groups at similar scales. So in the future, there's a couple things I want to do. First of all, I really want to complete the full fern tree of life and fill in that missing 50% of species we don't have yet. To do that, I don't think we can just wait around for sequences to show up in GenBank. We'll probably have to do some active sequencing, most likely of herbarium specimens, maybe via plastum skimming, which should work nicely with our existing plastum dataset. Furthermore, we want to integrate FTOL with the upcoming Pteridophyte phylogeny group 2, the new version of PPG. And that way, PPG will also be an online living document that can be continuously updated to reflect the most recent understanding of fern phylogeny. And finally, at some point, we want to transition to phylogenomics for all species. We're currently using Sanger data because that has the most species representation, but we do want to transition to phylogenomics going forward. And with that, I would like to thank all of the individuals and institutions who made this possible, and you all for listening. Thank you very much.